So we're doing the nervous system, chapter 11 right now. So first off, the function of your nervous system, the gist of it is going to be three things: sensory input, integration, and motor output. So your sensory input would be some sort of information being taken in and being brought to the brain. The integration would be the brain interpreting that, and the motor would be whatever your body's reaction to that sensory input is. So for example, I could touch a hot stove and my sensory input would be the searing heat and the damage to my tissues, and my brain would perceive that as pain, and my motor output would be a reflex to quickly pull my arm away. With the nervous system, it's going to be broken up into two parts. You're going to have your central nervous system and your peripheral nervous system, and we'll have a separate lecture for each of these guys. Your central nervous system is going to be the brain and the spinal cord, and that is going to be the command center. That's where integration is going to occur. That's where thought occurs. The peripheral nervous system is going to be where your sensory input and your motor output occur. Within your peripheral nervous system, you're going to have paired spinal and cranial nerves. Like we said, they're having the input and the output, so they'll carry information into and out of the central nervous system. The sensory division is afferent and the motor division is efferent. So sensory nervous system carries information into the central nervous system. Motor division carries information out of the nervous system. And I remember that this is always going to be the same, S-A-M-E. So sensory afferent, motor efferent, S-A-M-E. It's always the same, never changes. Sensory is afferent and motor is efferent. So looking at the peripheral nervous system, there's going to be two breakdowns of this. There will be the somatic nervous system and the autonomic or automatic nervous system, as I think of it. Somatic is under somatic, I think somatic muscle or skeletal muscle, so that's going to be under conscious control. That'll be all your skeletal muscles. The autonomic nervous system is going to be everything else. It'll be your nerve, it'll be your visceral motor fiber, so your smooth muscle or cardiac. It'll help regulate smooth muscle and cardiac muscle. And it's going to be broken up into two main divisions: your sympathetic division and the parasympathetic division. Sympathetic division, I think fight or flight. Parasympathetic division, I think rest and digest. So let's go through this breakdown again. We have our central nervous system, which consists of our brain and the spinal cord. You have your peripheral nervous system, which is always the same: sensory afferent, motor efferent. So sensory is going to be carrying information into the brain, and that'll be stuff like your organs, your stretch receptors and muscles, your stomach, stuff like that. Your motor is going to be broken up into two sections: the somatic, which is under conscious control, which will be your skeletal muscle, and your autonomic or involuntary nervous system, which is under subconscious control. And your autonomic nervous system is broken up into the sympathetic and parasympathetic nervous systems. Sympathetic is fight or flight. Parasympathetic is rest and digest. So think about it. When if a bear is going to come at you and eat you, what is going to happen? What's your reaction going to be? Are you going to sit there and just sit around like a mope? Or are you going to start breathing heavy, heart rate increases, you're getting ready, you're sending blood to all your skeletal muscles so you can run away and stay alive for another day? Parasympathetic, when I think of that, on the other hand, I think Thanksgiving dinner, you ate some turkey, you had a couple of drinks, you had some pie, you're way over full, and now you're sitting around watching football or something, and you're just on the couch, just you're relaxing. Your body is sending lots of blood to the digestive tract to take in all the food and take all the nutrients and sugars from all the food you just ate. And there's no point in having blood flushing the muscles right now because your skeletal muscles right now because you're not using them. You don't need them. So you're just relaxing. Okay, with the histology of the nervous system, we're going to have a couple different things going on. We're going to have neurons and we're going to have neuroglia. Neurons are going to be, when you think of nerves, neurons are going to be the cells that are going to make up our nerves. They're going to be the cells that are going to be excitable, and they're going to send electrical signals. So they will send information from one area to the next. Your neuroglial cells, or glial cells, those are your support cells. You're going to have some that are found in the central nervous system specifically, and some that are found in the peripheral nervous system specifically. The ones in the central nervous system are astrocytes, microglial cells, ependymal cells, and oligodendrocytes. And then our peripheral will be satellite cells and Schwann cells. So let's take a look at these guys here. Astrocytes are going to be the most abundant. When I think astro, I think asteroid, or I think out in the stars, so this is kind of like star shaping is what I think of. It's going to hold on to a bunch of nerves and capillary beds, and it's going to help guide nerves through their growing process when they are young. It's going to make sure they grow in the correct direction. Microglial cells, these guys are going to help with immunity in the nervous system. They will be around to phagocytize microorganisms and junk that is around. If there's ever a injury, they go over and they help clean up all the junk that is caused by the injury. So if you if you break a piece of a nerve, they need to clean up all the injured tissue before you can put down new clean tissue. Ependymal cells. These cells are going to be anything from flat and squamous to columnar cells, and sometimes they're ciliated. The big thing that they do is they will line the cavities of the brain and spinal cord. So they are going to separate the cerebrospinal fluid from interstitial fluid. Okay, next one is oligodendrocytes and Schwann cells. These guys are going to be responsible for myelination. With your nerves, you have two types of nerves. You have myelinated nerves and unmyelinated nerves. Myelinated nerves appear white, unmyelinated nerves appear gray. Myelinated fibers will carry information very, very quickly. Demyelinated fibers or unmyelinated fibers will not. Schwann cells are found in the peripheral nervous system. Oligodendrocytes are found in the central nervous system. Schwann cells will only myelinate a single neuron or a single nerve fiber. Oligodendrocytes will myelinate numerous nerve fibers. One of the theories is that Schwann cells are very important to regeneration of peripheral nerves, and the way I think about it is if I just have one cell and it's only taking care of one nerve fiber, if the nerve is damaged, the cell is going to want to help repair it and help everything grow. With this oligodendrocyte, if this nerve gets damaged and we break off this process right here, all of a sudden it's not attached to it anymore, it forgets about it, so it doesn't get myelinated anymore, and it's just gonna be demyelinated or die off. Okay, so our nerve cells, or our neurons, these are going to be the guys that are going to carry the electronic information, and these guys do not really regenerate. Very rarely they will regenerate. They can heal themselves sometimes, but they very rarely regenerate. What you're born with is what you get, basically. Not what you're born with is what you get. Once they're there, you don't usually grow very many new ones. They can live a long time. They can live up to 100 years or even more. However long you live is how long your neurons can live. They have a very high metabolic rate, and they need lots of oxygen and glucose. So if you stop breathing or they stop the glucose delivery, the cells can die. So here's our neuron right here. It's going to have some parts. It'll have its cell body, aka the soma. It'll have dendrites, which are these little short appendages coming off. It'll have an axon hillock, which will attach to an axon. And the axon will be covered by a myelin sheath. So cell body is going to contain the nucleus. This will be the command center. This is where all the thought process happens in the cell. Dendrites are going to be taking in information from the outside world. And the neuron, or the axon, is going to be the part of the cell that sends out a large electronic burst. Okay, dendrites and axons. Axons are long, alright? They're going to have knob-like terminals or buttons at the end. The thing with those knob-like terminals or buttons is going to be, that's where these guys are going to talk to each other through. They'll have these little synapses where they talk to one another. We'll see those in a little bit. If you have bundles of axons in the peripheral nervous system, you call it a nerve. In the central nervous system, we call them tracts. Okay, so myelination of peripheral nerves. Here's my Schwann cell. You remember Schwann cells were the cells that make the myelin sheath in my peripheral nerves? So you get your Schwann cell, and what it does is it starts to wrap around itself. And as it wraps around itself, it wraps so tightly that all the cytoplasm gets pushed out to the outer layer of the Schwann cell here. That outer layer is called the neural lemma. So if you think of cytoplasm, think of it as being like water. So this cytoplasm wrapped around the cell acts as water. Does electricity travel quicker through water or through dirt? I think we all know if you stick electrical cable in the dirt, you're probably going to be much safer than if you stick it in the pool that you're sitting in. So this is going to allow the electrical impulse to travel very, very quickly down the nerve. We have these things called nodes of Ranvier. I'm going to go back a couple slides to show you the nodes. Each one of these guys is a Schwann cell, or a node of a Schwann cell. All right. Between them, you're going to have nodes. So what happens is you go from neural lemma to neural lemma to neural lemma. When you're sending information, it kind of skips along these neural lemma. All right, it skips over the nodes. Rather than just walking slowly, if this was demyelinated, information would slowly walk along here. Since this is myelinated, it runs and skips from one neural lemma to the next. And we'll go on to talk more about that. Okay, myelin sheaths in the central nervous system is going to be formed by our oligodendrocytes, and we'll have our nodes, and these guys will also be myelinated. So we have white matter and gray matter. White matter is the myelinated fibers. Gray matter are unmyelinated fibers. Unmyelinated fibers are good for thinking and processing information. Myelinated fibers are good for quick actions. So somebody who is really, really good at math and good at just sitting there and staring and doing a single task would have lots of gray matter. A really good athlete, or somebody who's very dexterous and could do lots of physical stuff very well, would have a lot of white matter. People who could do both are lucky, I guess. I don't know. Okay, with our neurons, they could be shaped based off of what they do or how they look. So you have multipolar, bipolar, and unipolar. Multipolar will have lots of appendages coming off of them. Bipolar have 2 appendages coming off of them. And a unipolar has 1 cell body with 1 appendage coming off of that cell body. With our classifications of neurons, remember, it's always going to be the same. Sensory, afferent, motor, efferent. So sensory nerves will transmit information from sensory receptors to the CNS. Motor nerves will carry impulses from the CNS to effectors, which could be skeletal muscle, it could be other nerves, it could be glands, it's whatever the nerves are affecting. Then there's interneurons. Interneurons are going to talk to one another inside of the central nervous system. So this is where thought process occurs. Okay, so multipolar neurons. Okay, these guys are usually going to be our interneurons. A good example of these would be like a nerve going to a muscle fiber. These are long nerves. Bipolar neurons, and a good example of these would be your special senses. So, for example, vision are going to be using bipolar neurons. And then unipolar neurons, these guys are going to be first order sensory neurons. Okay, so functions of neurons. They're highly irritable. They will only respond to an adequate stimulus by generating an action potential. So if there's enough irritation, the nerve will be stimulated and it will have an action potential. The action potential is always going to be the same regardless of the stimulus. We call this all or nothing. So if the action potential goes, the nerve fires 100%. Therefore, let's say you're getting some pain. If you have like just a little bit of pain, maybe the nerve is firing 5 times per second. If you have a lot of pain, maybe that nerve is now firing 100 times per second. But the nerve is not firing any stronger, it's just firing more often. The frequency has increased. So some simple principles of electricity. Opposites attract. We take positive and negative charge magnets, they come together. Okay? We need energy to separate opposite charges. So if I'm holding two magnets and they're trying to come together, it takes energy for me to hold those together. If I let them go, that energy is released. So the energy caused by me holding them apart is called potential energy. When I let them go and they snap together, that energy is released. Okay? So that is kinetic energy. Some simple definitions. Voltage is just going to be a measure of the potential energy. Potential difference will be the voltage. Current will be the flow of electrical charge between 2 points. Resistance will be what is hindering the flow of electricity. So gray matter would have a high resistance to electrical flow. White matter would have a low resistance, so the electricity can flow much quicker. Insulator, it'll have high electrical resistance like gray matter. Conductor will have low electrical resistance like myelin. Okay, so we have our membrane ion channels. We're going to have proteins that are going to have two things. They can be leakage or they can be gated. Leakage means that we're going to be letting chemicals pass in and out of our nerve cell freely. Gated means there's a gate there and you got to open it up somehow. The 3 ways you can open it up will be chemical, voltage, or mechanical. Chemical gated channels will open by binding with the neurotransmitter. Voltage will be based on if there's a certain electrical charge that's going on, and mechanical will be based on some sort of physical deformation. Alright, so our gated channels, when they're closed, they're not going to let stuff pass in and out. Alright, so here's an example of a closed chemical channel. When it's closed, it doesn't let anything pass in and out. When a special neurotransmitter comes in, interacts with this receptor site here, it's going to let stuff flow in and out. So here we're letting sodium and potassium flow in and out of our nerve cell. This one is a voltage gated channel, so once the cell gets a certain voltage or charge, this channel opens up. Right here, this opens up, and we let sodium flush into the cell. So we're going to have potential difference across the membrane. In a resting nerve, the potential difference is going to be negative 70 millivolts. That will be our voltage that will be created. The way it's going to be created is there's going to be potassium inside of the nerve cell and sodium outside of the nerve cell. We are going to pump 3 sodiums out and 2 potassiums in. These are both positively charged, but since we're pumping 3 sodiums out and 2 potassium in, we get a general difference of a +1 charge with every pump. So, like we said, 2 potassiums go into the cell, 3 sodiums go out. 2 potassiums out, alright, 2 potassiums in, 3 sodiums out. So that's going to cause a difference in charge. The outside of the cell will be more positive than the inside of the cell, or the inside of the cell will be more negative than the outside of the cell. Okay, with membrane potentials, those guys are going to change based on the ion concentration, based on where sodium and potassium lie. Since we have more sodium outside of the cell, the outside of the cell is more positive. We have less potassium inside of the cell. That's going to make the inside of the cell more negative. With the changes in the potential, so we'll have 2 types of signals. They can be graded potentials, or they can be action potentials. Graded potentials are going to be occurring in dendrites. Action potentials are going to be occurring in axons. Graded potentials can change in their strength. Action potentials are that all or nothing. It goes or it doesn't go. It's 100% or it's nothing at all. These guys can build up to form action potentials. So with our changes in membrane potential, there's some different stuff that can happen. There's depolarization, there's repolarization, and there's hyperpolarization. Depolarization means that sodium is usually flowing into the cell, and the cell is going to, and the charge is going to become, or the voltage will become more positive. Repolarization is moving back towards our negative 70 millivolts resting potential, and hyperpolarization is going more negative than the resting potential. So hyperpolarization, an increase in membrane potential away from 0. So we're going away from 0. Alright, it's past our midline. Okay, so our graded potentials, these guys are going to be short-lived. There'll be changes that are in the membrane potential. Okay, so our graded potentials, those guys will spread as local currents, and they'll change the membrane potential, and those guys can build up to form an action potential. They will occur when a stimulus causes a gated ion channel to open. So some sort of like pressure or a receptor or a neurotransmitter will interact with that nerve, and it will cause gated channels to open. The more gated channels that open, the more likely you are to let that turn into a action potential. These guys will decrease in magnitude with the distance as they flow and diffuse through leakage channels, and they're going to have short distance signals. Okay, so an action potential is going to be when the membrane potential goes from negative to positive. Its amplitude is going to be about 100 millivolts. So since we start off at negative 70 millivolts, this moves into the region of about positive 30 millivolts. It'll occur in muscles and nerves, and it does not decrease in magnitude over distance. As opposed to graded potentials, these guys only travel a short distance, and they can decrease in magnitude over distance. These guys occur mainly in dendrites. These guys occur mainly in axons. So with your action potential, it'll have 4 parts: resting state, depolarization, repolarization, and hyperpolarization. Resting state sitting at negative 70. Depolarization is moving towards positive. Repolarization is moving in the negative direction. And hyperpolarization is when you move away from 0 past your resting state. So depolarization is going to be a reduction in potential across the cell membrane. Okay, so here's what happens. Here's our little video. Sodium flushes into the cell here. Alright, our charge goes positive inside, negative outside. When it propagates, this right here is just happening at one section. So this would represent a nerve cell. It happens from one section to the next, to the next, to the next, and it kind of walks along the nerve cell. So in a demyelinated or unmyelinated nerve, this would be occurring all the way down the nerve. In a myelinated nerve, it can like skip from one to the next to the next, so it can skip a bunch of these, skip super far along. So here's what will happen: positive to negative, and it goes right along the way here. Okay, you get the point. So our steps of an action potential: get depolarization in a single spot, the voltage will change and the channels will open up, and sodium ions flush into the cell. When those flush into the cell, the adjacent sodium channels open, and it just keeps working its way down and down and down. Alright, so we start off with a localized stimulus. It causes depolarization, and boom, it just shoots down the cell. And this is just kind of showing it right here. Okay, so your threshold. So with our axons and our nerves, we have our all or nothing. So here's our threshold. If you hit threshold, you're going to get an action potential. If you don't hit threshold, you don't get an action potential. Kind of like if I roll a marble up the side of an inclined plane or a ramp. If I get to the top of it and I don't pass it, the marble's just going to roll back down. If I get to the top of it and I go too far, it's going to fall. When it falls off the cliff, it has a set distance, is going to fall. That would be my action potential. Okay, so if I push it, it'll go. If I push it super hard off the inclined plane, it'll still fall the same distance. With the nerve, when you hit threshold, it fires. You get an action potential. If you have a greater, from your stimulus. If you have a greater stimulus, the only thing that occurs is the action potentials fire quicker. A higher rate or frequency is what will occur. Finally, we're going to get repolarization. So with repolarization, sodium channels close and potassium rushes out of the cell. Alright, so we get a resting memory potential, which is 70. We get our depolarization. When some sodium channels open, then boom, all the sodium channels open. That means that we hit membrane potential. And then we get our repolarization. Sodium channels close, potassium rushes out of the cell, and we go into our state of hyperpolarization right here. And our potassium channels close. Alright? The whole purpose of repolarization is to reset the nerve so it can fire again. It's not going to restore resting ionic conditions. Alright? The resting ionic conditions are going to be restored by our sodium-potassium pump. And what's going to happen, we'll pump in 2 potassiums, pump out 3 sodiums. So we'll have this thing called a refractory period. There's an absolute refractory period, and there's a relative refractory period. The absolute refractory period occurs during depolarization and the beginning of repolarization. What does that mean? The nerve cannot just fire and stay fired for a long time. It needs to begin to repolarize, and once the repolarization has began and the potassium is left, boom, it can fire again, and it can fire again, and it can fire again. So this relative refractory period means that the nerve is not going to fire unless it has a large stimulus. Absolute means the nerve cannot fire until the potassium has began leaving the cell. Okay, so once again, our relative refractory period. Most sodium channels have returned to their resting state. Some sodium channels are still open, but the nerve can fire again now because we have the beginning of a potential difference again. Okay, so we got this stuff called conduction velocity. Conduction velocities in nerves vary widely. If nerves are wider, it can depend on the nerve width, the diameter of the nerve, or it can depend on the myelination. So if the nerve is myelinated, it's going to conduct a lot quicker. If it's demyelinated, it'll conduct a lot slower. If the nerve has a larger diameter, it will carry information quicker. If it has a smaller diameter, it'll carry information much slower. Okay, so with our unmyelinated nerve, it's got the bare plasma membrane, and it travels very, very slowly down the nerve. Alright, with our myelinated membrane, it can skip along from node to node to node to node to node, and as it's skipping along, then that nerve is going to be able to travel much quicker down the nerve. So multiple sclerosis, you guys have heard of this before. This is an autoimmune disease, and what multiple sclerosis does is it affects the myelin sheaths in the central nervous system. So what that means is that the nerves in the central nervous system cannot talk as quickly. They're going to be able to fire normally, but they're not going to be able to conduct and speak with one another at a rapid pace. So next one, we're at the synapse. This is going to be where 2 nerves meet. You have a presynaptic neuron, a postsynaptic neuron, and between the two we have a synaptic cleft. Alright? Presynaptic neuron is going to send information towards the synapse. Postsynaptic neuron is going to take information away from the synapse. So we'll have two types of synapses, electrical and chemical. Electrical synapses aren't very common, but they'll be really, really nice because they can talk very quickly and they can go either direction, forward or backwards. Chemical synapses, when you think of a normal synapse and you think of a muscle, this is a chemical synapse. These guys are going to be one directional, alright, and you'll have an axon terminal, which is going to be presynaptic, and you'll have a receptor region, which will be postsynaptic. It can be another nerve, it could be dendrites, it could be a couple different things. So here's what we were looking at here. Here's your axon terminal. This is presynaptic. Here's your synapse. And here's your postsynaptic. So what happens? An action potential travels down my nerve. The voltage gated channels in my axon terminal allow calcium to flow in. When that calcium flows in, we begin to release these synaptic vesicles. These are filled with neurotransmitters. Those guys cross the synapse. They interact with these proteins, and when the neurotransmitter touches it, it opens it up, and it's going to allow flow to occur, and that will help us begin our next step of the nerve firing. So that'll be the way the nerve goes from this nerve, the action potential goes from this nerve to the next nerve. So action potential arrives at the axon terminal, calcium comes in. It tells us to release our vesicles, the neurotransmitters travel across the synapse, the ions bind with their proteins, the proteins open up, and then we're going to have an enzyme released to break down all of these enzymes. Why? So the nerve doesn't keep firing. Okay, so how are the neurotransmitters going to stop? Like we said, they're broken down by enzymes, and they get reuptaked. Why do we reuptake them? So we don't need to make new ones. You're just being efficient. Synaptic delay. This is going to be the rate limiting step of your nervous system. This is what is going to slow you down when you have some sort of like reflex, or why your reaction time is not just immediate. It takes a split second. The average synaptic delay time is about 0.3 to 5 milliseconds. It's very, very, very short. So we'll have our postsynaptic potentials. These guys will be our graded potentials. These guys will be determined by the amount of neurotransmitters and the time the neurotransmitter's been in the area. The longer it's been there, the more stuff can flow into the cell, so it'll be a stronger grade. There's two types. There's excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Excitatory would do stuff like make you more jittery. Inhibitory would do stuff like slow you down. Okay, so let's compare graded versus action potential. Graded potential is going to typically occur in our dendrites. Action potential will happen at the axon hillock and travel down the axon. Graded potentials are short. In these dendrites, action potentials go long. Sometimes they could go a meter or even further. Graded potentials have graded or different sizes. Action potentials, all or nothing. It's always the same. It's always about 100 millivolts. With our graded potential, they can be chemical or sensory stimulus. Action potentials are only going to be voltage. Graded potentials are going to have an absent positive feedback cycle, and our action potentials will have a positive feedback cycle. And then these guys, they don't need to always repolarize. These guys need to repolarize. Okay, going on. Stimuluses. Okay, you can have a temporal or you can have a spatial. Temporal means you have more stuff coming in over time. The frequency is increased. Spatial means you have a bunch of dendrites getting activated. Alright, doesn't occur in an action potential. With our EPSPs and IPSPs, alright, our EPSPs, that's going to increase the amount of information that can come into the nerve cell, so it'll be easier excited. This will decrease the information. So this could be something like caffeine, and this could be something like Benadryl that puts you to sleep. Alright, EPSP makes your resting membrane potential move closer to threshold, which means it'll be more jittery. And IPSP makes your resting membrane potential move away from threshold, which means it will be less likely to fire. Okay, so integration and summation. How does this stuff work? An EPSP alone cannot make an action potential occur. If you put a bunch of them together, it can get you to really reach threshold, and an action potential can occur. If you have EPSPs and IPSPs occurring at the same time, they're just going to cancel each other out. And we already talked about temporal or spatial. So temporal means the frequency of the graded potentials are increasing or decreasing. Spatial or summation means that all of the dendrites are getting activated rather than just one. Alright, with repeated use, increases the efficiency of a neurotransmitter. What do I mean by that? First time you go try to shoot a basketball or swing a golf club or drive a car that stick shift, it's very, very hard to do. The more you do it, the easier it gets. So you ingrain a pattern in your nerves. Your nerves get used to this activity and they get better at it. So when you try something once and you fail, don't stop. You keep doing it over and over again. Why? You get better. You can learn. So next we're talking about our neurotransmitters. There's over 50 neurotransmitters that we know of, and I'm sure there are many that we don't know of. They are classified based on their structure and their function. So here's the categories we're going to want to talk about. Acetylcholine. This is going to be real important for muscles, the neuromuscular junction. You remember we talked about this in the muscles chapter. And a little bit in the autonomic nervous system. ANS is autonomic nervous system. We get our biogenic amines, alright? Those will be our catecholamines and idolamines. When I think of catecholamines, I'm always thinking epinephrine and norepinephrine. These guys are going to be our sympathetic, these guys are going to help with our sympathetic nervous system. We get our peptides, which could be like substance P, endorphins, alright? We get other peptides like somatostatin and cholecystokinin. Then we get our amino acids. Okay, we get novel messengers, so we could get like purines or gases, or we could get carbon monoxide, and we could get our endocannabinoids. So different neurotransmitters have different actions. Some of them can be excitatory, some of them can be inhibitory. If they're excitatory, they cause depolarization. If they're inhibitory, they cause hyperpolarization. And it doesn't matter on what the neurotransmitter is, it matters on the location the neurotransmitter is. For example, acetylcholine causes excitation in neuromuscular junctions and skeletal muscles. If you put that in the cardiac muscle or in the heart, it's going to cause inhibition, so it's going to make it not want to contract. Okay. With their actions, they can be either direct or indirect. Direct action means it'll go like right to a channel and it'll open up the channel. Okay. Indirect will mean it'll do something almost like a hormone. It'll have a longer lasting effect. It'll affect everything around it to make the cell do what it wants to do, excite or inhibit. And that's all.